The PREDICELL Uptake Transporter Kit from SOLVO Biotechnology is a ready-to-use kit with pre-plated live cells, buffers and reagents, enabling you to conduct in-house drug uptake transporter studies. Upon receipt of the PREDICELL kit, carefully unpack the contents. Place the buffers provided in a 37°C water bath to equilibrate. Carefully remove the pre-plated cells from the package. Remove the plate from the vacuum bag and examine the cells under a microscope to ensure that they have not been damaged during transit. The cells should then be placed into a 37° incubator for 2 hours. Alternatively, the plates can be stored overnight. However, aseptic media replacement and a CO2 incubator is required. Prepare a serial dilution of the test compound or reference inhibitor on a 96-well dilution plate. Prepare the probe substrate solution in transport buffer, containing a trace amount of radiolabeled substrate, and supplementing with unlabeled compound to achieve the desired final substrate concentration. Mix well and aliquot the probe substrate solution onto the auxiliary plate. Add the serial dilution of the test compound, as well as vehicle control and reference inhibitor, to the substrate solution on the auxiliary plate and pre-incubate at 37°C. Retrieve the PREDICELL plate from the incubator and aspirate media from the cells using a vacuum manifold, taking care not to disrupt the monolayer. Wash the cells with pre-warmed transport buffer. Add pre-warmed transport buffer and return the PREDICELL plate to the incubator for 15 minutes. Retrieve the PREDICELL plate from the incubator and arrange it in the same orientation as the auxiliary plate. Aspirate the transport buffer from the wells and dispense the appropriate solution from the auxiliary plate onto the PREDICELL plate. Place the PREDICELL plate at 37°C for the required incubation time. After the incubation period, aspirate the solution from the wells and immediately wash with ice-cold transport buffer. Aspirate the transport buffer and lyse the cells by adding the cell lysis solution. Incubate at 37°C for 10 minutes. Analyze the samples using liquid scintillation counting, making sure to also measure the total counts in the probe substrate solution.